from Quezon City, Philippines. Your news, current affairs, and public service channel.
A human baby without a mother and a father to help. But they have made a striking progress in showing an embryo basically from scratch.
This is what a 2-week-old embryo looks like, but it didn't come from the fusion of a sperm cell and an egg cell. It was made from stem cells. Researchers from the Weizmann Institute of Science in Israel learned how to reprogram stem cells into cells found in an early-stage embryo. After Jacob Hanna's team mixed them together, a few turned into balls of cells called aggregates, which grew into something that strikingly resembled human embryos.
About 1% of the aggregates, we can see that cells start differentiating correctly, migrating and sorting themselves into the correct structure, and the farthest we could get is day 14 in human embryo development.
Scientists in the field are quick to point out that even if the cell aggregates start to look and act like embryos, they're still far from the real thing. The method has limits.
I want to emphasize that if you're talking about trying to make a whole baby pregnancy outside the uterus, that is just impossible, because the human embryo is very big. Pregnancy is 9 months, so that is not. It's not our goal. It's actually also impossible, and there are no concerns about that at all.
Instead, the goal is to create models that give a better understanding of what happens directly after an egg is fertilized, without the ethical concerns involved in using real human embryos. Even now, too little is known about early development and what can go wrong in the first crucial weeks after conception.
It's a time when many pregnancies fail. So a fresh insight that might come from the use of these stem cell-based models of human embryos might lead to a better understanding of the causes of miscarriage, and indeed some of the really unique aspects of these early stages of human development.
That could lead to important medical breakthroughs in reproduction, but the technology also takes us one step further into an ethical minefield.
And let's get more on that. We are joined here by DW Science reporter Anna Carthouse, who's on this story. It sounds like a potential game changer. How excited are you about this research, or are you concerned?
Well, Sarah, the thing is, we know disturbingly little about the beginning of our existence. Our first days and weeks of life are impossible to study without compromising the safety of the embryo in the womb. But at the same time, this is a period in which a lot of pregnancies fail, and we just don't know why. So embryo models are an amazing and really striking workaround to study this period in this black box. They could help and treat miscarriages, genetic diseases, or birth defects. They could be used to probe the safety of drugs used during pregnancy, or they could be used one day, but that's really a future scenario, to grow tissues and organs for surgical transplantations. So one could say, as a science nerd, I'm pretty excited.
So why is it such an ethical minefield then, if it does have those positive potential practical applications?
Sure, so to study these questions, which is a laudable endeavor, scientists want to come as close to real human embryos as possible, as they cannot do research on the embryos themselves. And until recently, that wasn't a problem, as the embryo models they created bore only a sketchy resemblance to real embryos. Just these times are over now. One stem cell biologist told me that seeing these embryo models sent a chill down his spine, as they were so similar to natural embryos. That's why researchers are eager to say, these are not real embryos, these are not real embryos, these are just models. But how do we say, this close is too close?
And you know, the scientist we have to mention in that report says that there's still a long way from recreating human embryos. So if we're honest though, I mean, is that the ultimate goal?
Yeah, the models created by the groups of Jacob Hanna and other groups earlier this year are incredibly similar, but they're not identical with human embryos. And at the moment, that's not the goal, not for the scientists. They want to understand how the processes work. They don't want to create artificial life. And there are also legal restrictions preventing that. And these embryo models are far from the stage from being transplanted into the womb, which is what we would need in order to nourish them. It's possible to nourish a couple of or a cluster of cells in the petri dish, but doing this with a baby becomes pretty advanced. So it's not the goal at the moment, it's pretty far away, but research is progressing at an unprecedented speed. So at the moment, there is no human or animal embryo model that ever formed a neonate, but science is taking baby steps towards
what's your big takeaway then? How should we all feel about this?
I'm torn apart. It's on the one side, it's really a major milestone in science. On the other hand, it brings humanity closer towards something we're really taking our hands off from. We don't want to create artificial life, and the research community and society has to ask this question: where do we say halt to this research, and where do we say this is what we need in order to understand our early days and understand basic processes, but this is kind of a step too far.
Okay. DW science reporter Anna Karthaus putting this all into context for us. Thank you so much.
Lingkod bayan sa'yo'y nakalaan. Narito kaibigan ko, palad namin alay sa'yo, sa gitna man ng unos at tag-ulan.
Okay, maraming salamat sa pagkinig mga iniirog ko mga Pilipino. Nandito naman po ang inyong lingkod, Doc Ancient Specialist Alien Medico, para po sa ating pag-aaral ng stem cells. At nakita po natin kung paano ang embryonic, yung early stage of human creation, ay hindi po talaga pwedeng mag-create ang ating siyensya ng biology at scientist ng another life in artificial way. Pero ang cloning, mga kaibigan, merong tinatago ang ating technology na hindi ko pa rin po lubos na maunawaan. Ang challenge po ng ating behavioral biologist and scientific is almost ending to failure of trying to search the best error of limitations of their intellectual body, kung saan ay yung pong pluripotent stem cells ay talaga namang napakahirap mag-replicate. Kahit kunin mo pa yung early stage of human development, yung mga embryonic niyan, yung mga cells ng mga eggs sa sperm cell ng human being, ay hindi po talaga makakagawa ang scientist ng living things na tulad ng ginawa ng ating mahal na Panginoon. Wala na po yun talaga, mga kababayan. Pero yan po ay may natatagong isang bagay na hindi ko pa po lubos na maunawaan sa kanilang pag-aaral. So kung ano man po yun, ay dapat po natin na abangan kung paano kakaroon po ng natural cloning ang synthetic stem cells, Ayan po ang pag-aaral kung saan po ay talagang naluha ang mga dalubhasang scientifico biologists sa impact po ng erroneous na pagkakaroon ng engineering system na hindi talaga maunawaan, kundi maghanap pa po ng another development programming for such kinds of deadly diseases. Kaya po yung aming case of death, mga kaibigan, yan po ang regenerative medicine ng natural ng ating nature. Kaya po ito ay talagang hindi po kayang aralin pa ng kahit na Estados Unidos na ikay mawalan ng hininga ng isang oras. That is so much deadly, Pilipino. Kaya yung mga legal factor po niyang pagkagawa ng ganitong creation ay may mga legal sanction na sinasabi na bawal ang mga scientists na gumawa ng ganitong pag-aaral. Pero nag-exist na po yan, may cloning na nga eh. Kaya lang medyo yung results po ay tinigil at sinabing hindi raw nakagawa ang kanilang siyensya ng biology ng another living organism sa pamagitan ng artificial discovery ng ganitong stem cells. Yung mga embryonic po nito ay talagang napahirapan ang mga siyensya. Yung mesenchymal stem cells, plurimin TENTA stem cells, pinahirapan po sila. Amen.